Welcome to G Fun Facts Online. Today, we are stripping away the medical jargon to look at the mathematical chaos hiding inside the human body. We are going to talk about a revolution happening in oncology, one that isn't about biology in the traditional sense, but about physics, economics, and hard mathematics. We are talking about how we predict the unpredictable behavior of cancer. Picture this scenario. You are in a hospital. It is quiet, sterile. A patient looks at their oncologist and asks the most fundamental question. Doctor, will this treatment work? It sounds like a simple yes or no question. But for decades, the answer the doctor gives has actually been a statistical guess. It is a probability derived from the average outcomes of thousands of other people. But here is the problem. Cancer is not an average. It is a rogue biological entity. It is a chaotic system that evolves, adapts, and behaves with a terrifying degree of unpredictability. Standard medical models have been built on the assumption that biology follows a straight, deterministic line. They assume that if you hit a cell with drug A, it will die. But what if the cell doesn't die? What if it waits? What if it retreats, changes its camouflage, and returns stronger? This is where the frontier of stochastic biology enters the fray. We are moving from a war of attrition to a chess match against entropy itself. To understand why this is such a massive shift, we have to look at the illusion of order we have lived with for the last century. For over 100 years, oncology was dominated by deterministic thinking. The most famous example of this is something called the Gompertzian growth curve. This was a model developed way back in the 19th century by Benjamin Gompertz. Ironically, it wasn't designed for cancer. It was designed to calculate life insurance premiums by predicting human mortality. Later, scientists realized the math fit tumor growth. It is an elegant, simple model. It says tumors grow fast when they are small and slow down as they get larger and run out of resources. It produces a smooth, S-shaped curve. If you trust this model, you believe that you can predict the size of a tumor at any future date just by knowing its current size and growth rate. It is comforting. It implies order. But cancer cells do not obey smooth curves. They are subject to stochasticity, which is the scientific term for randomness. Think about gene expression noise. You can have two genetically identical cancer cells sitting right next to each other. They should behave the same, right? But they don't. Because of random fluctuations in which genes are turned on or off at any given millisecond, one might die from chemo, and the other might ignore it. This is the butterfly effect in biology. A single mutation in one single cell, occurring at a random moment, can spawn a resistant subpopulation that renders a therapy useless months later. The old deterministic models treat a tumor like a balloon inflating, predictable, uniform. Stochastic models, however, treat it like a swarm of bees. You can predict the general direction of the swarm, but the path of every individual bee is erratic, influenced by wind, predators, and the behavior of its neighbors. Ignoring this randomness is exactly why we see so many miracle drugs work in the petri dish, but fail in the human body. The petri dish is a controlled, deterministic environment. The human body is a chaotic, stochastic storm. So how do we solve this? We stop treating the randomness as noise to be filtered out, and start treating it as the driver of the disease. This is where stochastic biology comes in. Scientists are now using stochastic differential equations. Instead of smooth lines on a graph, these equations include a specific noise term that accounts for random biological fluctuations. They are using branching processes, math originally used to model nuclear chain reactions, to calculate the probability of a single resistant cell branching out into a fatal colony. By quantifying the unknown, these models don't just predict what will happen, they predict what might happen. They assign precise probabilities to different futures, allowing oncologists to prepare for the worst-case scenario before it even manifests. Now, let's get into the most fascinating application of this, game theory. You might know game theory from economics or political science. It predicts how rational humans behave when their success depends on the actions of others. Like the famous prisoner's dilemma. Well, it turns out, cancer cells are players in a game too. Dr. Robert Gatenby and his team at the Moffitt Cancer Center have been pioneers in applying this logic to oncology. They realized that a tumor is not a single block of tissue. It is an ecosystem of competing players. In this game, you generally have two types of players. Player 1, the sensitive cells. These grow fast, they are aggressive, but they are vulnerable to chemotherapy. Player 2, the resistant cells. These are immune to drugs, but there is a catch. Because they spend so much energy maintaining their defense shields against toxins, they grow slowly. They are biologically expensive to maintain. In a natural environment where no drugs are present, the fast-growing sensitive cells are the bullies. They outcompete the resistant cells for food and space, keeping the resistant population small and suppressed. In game theory, this is called a Nash equilibrium. But then, we intervene. Traditional chemotherapy applies a strategy called the maximum tolerated dose. The logic is simple, Bombard the tumor with as much drug as the patient can survive to kill as many cells as possible. But game theory reveals the fatal flaw in this strategy. When you hit the tumor with a massive dose, you kill all the sensitive cells, you wipe out the bullies. But you leave the resistant cells behind because the drug doesn't hurt them. With their competitors gone, the resistant cells experience what is called competitive release. They suddenly have all the space and nutrients to themselves. They explode in number. The tumor returns, but this time, it is composed entirely of resistant cells. The drugs no longer work. That is checkmate. Using this insight, Gatenby's team proposed a radical new strategy called adaptive therapy. The goal here shifts. We stop trying to kill all the cancer immediately. Instead, we try to manage it. The strategy is to give a small dose of the drug, just enough to kill some sensitive cells, but leave enough of them alive to keep suppressing the resistant cells. The result? The tumor shrinks, but it doesn't disappear. The sensitive cells remain the dominant player, preventing the resistant supervillains from taking over. Because the sensitive cells are still there, the drug continues to work. And the proof is in the pudding. In a landmark clinical trial for metastatic castration-resistant prostate cancer, patients treated with this adaptive therapy, where dosing was based on an algorithm, not a fixed schedule, survived significantly longer than those on standard high-dose therapy. They used less drug, suffered less toxicity, and lived longer. The math didn't just predict the behavior. It outsmarted the evolution of the disease. Let's move from the tumor itself to the most dangerous aspect of cancer: metastasis. This is the spread of cancer to other organs, and it is responsible for 90% of cancer deaths. It is also an inherently stochastic or random process. Think about it. A cell has to detach from a tumor, survive the turbulence of the bloodstream, evade the immune system, and lodge itself in a bone or a lung. This is a rare event. It's like a gambler playing a slot machine. Most cells die. They lose the gamble. But if the tumor releases millions of cells a day, the laws of probability state that eventually, one will hit the jackpot. Dr. David Basanta, also at Moffitt, uses something called agent-based models to simulate this. Imagine a video game like SimCity. In Basanta's models, the agents are individual cancer cells, bone cells, and immune cells. Each agent follows simple rules. For example, a cancer cell agent might have a rule that says, if I meet a bone cell, I release acid to dissolve it and make space. Or, if I divide, there is a 0.001% chance I mutate. By running this simulation thousands of times, a technique called Monte Carlo simulation, researchers can see patterns emerge that are invisible to the naked eye. They discovered a vicious cycle in prostate cancer bone metastasis. The cancer manipulates bone destruction, which releases growth factors trapped in the bone, which in turn feeds the cancer. Crucially, the models predicted that hitting this cycle at the wrong time could actually make things worse. This mathematical insight helps doctors optimize the timing of bisphosphonate drugs to break the cycle without fueling the fire. This kind of modeling is also revolutionizing how we treat brain cancer, specifically glioblastoma. This is a deadly cancer known for being diffuse. Even after a surgeon removes the visible tumor, invisible individual cells have already drifted inches away into the healthy brain tissue, like ink spreading in water. Dr. Franziska Mashor at the Dana-Farber Cancer Institute uses stochastic models to track these invisible invaders. Her team uses reaction diffusion equations. The reaction part calculates how fast cells divide. The diffusion part calculates how fast they move through the brain's white matter highways. By analyzing a patient's MRI with these equations, the math can estimate a probability cloud of where the cells are hiding. This suggests that instead of standard radiation around the surgery site, we need to alter the radiation schedule to catch these moving targets. It is about calculating the geometry of recurrence. So where is this all leading? The ultimate goal of stochastic biology is something borrowed from engineering: the digital twin. In aerospace engineering, a digital twin is a virtual replica of a jet engine. Sensors on the real engine feed data to the virtual one. If the virtual engine predicts a failure in 500 miles, mechanics fix the real one before it breaks. In oncology, the virtual patient is being born. Here is how it works. The model is fed the patient's specific data, genetics, pathology slides, MRI scans, and crucially, single-cell sequencing data, old technology used to mash a tumor up and sequence the average DNA. Like blending fruit into a smoothie, you lose the detail. New technology called single-cell RNA sequencing looks at every cell individually, like a fruit salad. You can see the strawberry, the grape, the melon. This data allows stochastic models to see the heterogeneity, that rare 1 in a million cell that harbors the resistance mutation. The computer creates a virtual version of the patient's tumor. It doesn't just run one simulation, it runs millions, exploring every possible evolutionary path the cancer could take. The doctor can then wargame the treatment. They ask the digital twin, what happens if we give chemotherapy A? The model might reply, 80% chance of remission, but 90% chance of resistant recurrence in 8 months. Then the doctor asks, what if we alternate chemo A and immunotherapy B? And the model replies, recurrence probability drops to 20%. This is the future. We are standing on the precipice of a new era where biology becomes a data science. The war on cancer is transitioning into the calculation of cancer. This transition isn't easy. Biological data is messy and noisy, but the successes we are seeing in adaptive therapy for prostate cancer, in optimized radiation for brain tumors, prove that the concept works. Stochastic biology teaches us a humble lesson. We cannot eliminate chaos. Cancer, like life, is inherently unpredictable. But with the right mathematics, we don't need to eliminate the chaos to win. We just need to understand the rules of the game better than the cancer does. By embracing the randomness, by calculating the uncertainty, and by respecting the evolutionary intelligence of our enemy, we are finally learning how to stay one move ahead. Thanks for listening to G Fun Facts Online.